This is the presentation over the materials in Chapter 9, talking about growth and metabolism, subtitle running the microbial machine. Now, the fact is that the objectives that I have for you are considerably less than the objectives that are in your text. And if we go to this next slide, you'll see that this is really what I want you to know. You should understand the dynamics of growth curve for a microbial population and specify some of the influence that affect the shape of the curve. Recall some factors such as variations in water, temperature, and oxygen levels that encourage or limit the growth of certain categories of microorganisms. Discuss the broad diversity of microbes on Earth's environment and understand why that diversity is important for survival. What I want you to get out of this chapter are some materials that will help you understand a future chapter when we start talking about controlling microorganisms. This is one of the big objectives for our surgical technology program. We will actually spend quite a bit of information on control of microorganisms, so this is just a primer to get you ready for those materials later on. Now, before we can really talk very much about metabolism and growth and all of those other things, we have to define some of the processes of life. And we've mentioned some of this before. This is not in your text, but it's important to understand the nature of organisms. Processes of life include growth, and that can be growth in numbers, growth in size. Reproduction, that is definitely growth in numbers. Metabolism, all living things have activities going on, chemical activities. Movement, and that can just be movement in and out of a cell of materials, but it can also include irritability, that is the ability to respond to the environment. Additionally, we've already stated that all life is cellular in nature, so viruses are not considered living. All life, as we've said from Chapter 1, comes from pre-existing life. And all body processes are a product of chemical reactions. So all life processes, basically all of the chemical reactions, are the metabolism. What metabolism is it's all the chemical changes that occur in a microbe during growth and development. And there's two major categories. We have anabolism, which is something that builds materials. Generally requires energy, and I think of like anabolic steroids, the things that pump you up. Catabolism is breaking down materials, and in general this releases energy. Now, if I need to feed energy in to build something, then I usually am breaking something else down to give me that energy. So these reactions occur side by side. So when we talk about population growth, when we first introduce organisms into a new environment, there is a lag phase. They don't grow very much for a short period of time because they're adapting to their environment. Once they've adapted to the environment, there is a logarithmic phase where the cell numbers increase exponentially. You know, we start with 2, then we have 4, then we have 8, then we have 16, then we have 32, 64, and so forth. So this actually, that curve should steepen over time. But at some point, there is a balance that gets reached where resources that are available just meet the needs of the community. And that's our stationary phase, where we just have sort of a steady rate of, we don't have an increase in population. Now eventually, as we start to destroy the resources in the environment, what will happen is a decline or death phase, where the majority of the cells are going to die off because there's no longer the resources that they need in the environment. Usually, there are some cells within the environment that remain viable. And then if you look here, you can see we have, on this chart, we have the red signifying living cells, the brown signifying dead cells. And eventually we have this decline or death phase. So, growth curve. Lag phase is a flat period of adjustment, enlargement, very little growth. And then we have an exponential growth phase, or logarithmic phase, or log phase, a period of maximum growth which will continue as long as cells have adequate nutrients and favorable environment. There is a stationary phase, where the rate of cell growth equals the rate of cell death, and it's caused by the fact that there is a depletion of nutrients or oxygen, and also the excretion of organic compounds that act as pollutants. And then the death phase. As limiting factors intensify, as there are fewer and fewer nutrients and more waste products and pollutants, eventually the cells will die, also exponentially. They die off very rapidly, pretty much drowning in their own waste products. This is one of the reasons why a bottle of wine can only have about 7% alcohol. It's because at that point, the yeast organisms have polluted the environment with sufficient quantities of alcohol to actually have killed off the yeast. So this gives us kind of an idea of this population growth. And when we look at microbes, we talked about E. coli being able to double its numbers every 20 minutes. So imagine that at minute 1 we have 1 cell. 20 minutes later we have 2 cells, and 20 minutes after that we have 4 cells, and 20 minutes after that we have 8, and 20 after that 16, and 32, and so forth. And so this is the type of curve that we actually see. It is not a straight linear curve. So again, normal growth curve, lag phase, exponential growth, and that curve should be sharper, stationary phase, death phase. Aside from limited resources, there are other environmental factors that will influence microbial growth. If we have limited resources or even unlimited resources, the fact is that some organisms live in different temperature ranges. Some will have different oxygen requirements. Some survive in better pHs. Some actually live better in certain areas of radiation. Barometric pressure is a factor for some. Light and UV and other nutritional requirements can all come into play when in the growth of some organisms. There are temperature influences first. Most organisms have a minimum temperature that they'll grow. It's not the best, but they can survive. There's a maximum temperature, which is the highest temperature that permits growth and metabolism. Then there's the optimum temperature, where everything works just perfectly for them. 3 different ways that organisms have adapted. So if we go back here, minimum temperature for one organism may be the optimum temperature or the maximum temperature for another. And we have these types of organisms that like different environments. We have psychrophiles. These function best in cold temperatures. Optimum temperature for them is below 15 degrees Celsius, and some are capable of growing at 0 degrees. We're talking freezing. Mesophiles function best in what we would consider a normal temperature. Optimum temperature between 20 and 40 degrees Celsius, and that includes our body temperature. So this group of organism includes most human pathogens. The psychrophiles really aren't going to do that much to us because our body temperature is just too hot. Thermophiles prefer a hot temperature, and their optimum temperature is greater than 45 degrees centigrade. So they are also not a threat to us because they are outside of our temperature range. Here's our temperature adaptations, or adaptation groups. We can have minimum, optimum, maximum. So Temperature, water and temperature. Psychrophiles. These are things that grow nicely in your refrigerator. We also have facultative psychrophiles. Those are ones that will grow in cold temperatures, but they don't like it very much. They're capable of it. And mesophiles, thermophiles. There is also a group of hyperthermophiles. Now, an example of the mesophiles we see right here, intestinal gases formed by E. coli. We see moldy bread. We see that often when it's kept colder. And we see thermophilic algae that like to grow in hot temperatures. Other factors, oxygen level and acidity. Now, there are a group of organisms called obligate aerobes. That is, they have to have oxygen. Humans are obligate aerobes, must have oxygen to function. There are obligate anaerobes that cannot exist in the presence of oxygen. They have to have, their metabolism functions differently, and oxygen is actually poisonous for them. There are facultative anaerobes, and these live in environments with varying quantities of oxygen. They don't like oxygen at the level of the air that we breathe, and they're not anaerobes. They have to have some oxygen. So these tend to live just below surfaces, slightly isolated. We can see this in watery environments. Now, microaerophiles must have oxygen, but in really small quantities that exist in the atmosphere. And then there are aerotolerant anaerobes. They do not need oxygen, but can survive in its presence. Now, what happens with these organisms? You can grow these in tubes, actually, and toward the bottom, you're going to see very little oxygen, and at the top of the tube, quite a bit. So an aerobic organism is going to grow at the top of the tube. A microaerophilic, you're going to see a band of growth just below the surface. Facultative anaerobes, they can grow anywhere in there. Aerotolerant anaerobes prefer growing at the bottom, but they can survive somewhat. So you'll see growth throughout, but you'll see more growth toward the bottom. And anaerobic, what you're going to see, no growth at the top, growth of the organisms at the bottom of a tube. And this is one way we can evaluate the nature of microorganisms, is growing them in tubes just like this. Now, there are some microorganisms that have to have a zero oxygen environment, and what we do is put them in a chamber, lock the chamber down. There can be a gas generator envelope which is added, and they will carbon dioxide and hydrogen gases. Oxygen is removed from the chamber by combination with the hydrogen, and it reacts on palladium, which basically makes it form water. I've seen other chambers similar to this where you basically lit a candle, closed it up, and the candle burned until all the oxygen was gone. There are organisms that have different demands for the acidity in the environment. Those that like an acidic environment are acidophiles. Those that like a neutral pH, very close to 7, are neutrophils, and those that like an alkaline pH are alkalophiles. Other factors that can come in, salinity level. So there can be organisms that like to grow in water that has no saline. Those would be osmophiles. Those are things that are going to grow in pond waters and things like that. And then there are halophiles, which are organisms that are going to grow in the oceans. They can grow in salt lake. They can grow in the Dead Sea, depending on how tolerant they are or how much they like being in saline environments. organisms that have different levels of pressure that they can survive at. There are organisms that live in the deep sea that would simply burst if they were brought to the surface. They cannot survive outside of that very intense pressure range. Again, radiation can come into play. We use ultraviolet radiation to sterilize surfaces. We use gamma radiation also to sterilize. And X-rays can cause damage to organisms and kill them. But there are organisms that can survive in all of these. The pH factor. We talked a little bit about this in chemistry, but just as a refresher, a neutral pH is 7 on a scale of 0 to 14. Acids are numbers less than 7, and base have numbers greater than 7. And our own body pH is around 7.35 to 7.45. Now, most organisms live in a pH range between 5 to 8. There are fungi that are in general acidophiles, liking a pH of about 5 or less. On the surface of oranges, we have one bacteria that is an acidophile that is actually an extreme acidophile, which is Heliobacter pylori. And the pH in your stomach is pretty close to 1. And this microorganism appears to be the one that causes about 80% of stomach ulcers. Halophiles like high salt concentration, some as much as 27%, and many of these live in the Great Salt Lake. Barophiles like a high pressure, and they live in the ocean floors. UV normally kills bacteria, but some can resist radiation. Some, in fact, live in radioactive waste areas. They've specially adapted just for that. Many organisms have some specific nutritional needs. For instance, humans, there are 20 amino acids, of which 10 we cannot make, so we have to have those provided to us. The same with certain organisms. These organisms can only be cultured in very specific media. So there are some organisms that can only be cultured in a blood agar media, for instance. There are actually selective media that we use to separate out microorganisms or isolate certain organisms from others, and we use what we're doing is we are attending to the specific nutritional needs of some organisms while excluding the nutritional needs of others. Now, microbial metabolism. We talked about how we get energy, and we talked about anabolism and catabolism. There are metabolic processes that give microorganisms readily available sources. Photosynthesis, we've talked about cyanobacteria, we've talked about algae. There's also respiration, where we break down glucose and other carbohydrates to capture energy. And so when we're talking about this, we're talking about a heterotrophic type of lifestyle where photosynthesis is an autotrophic. The final product, regardless of photosynthesis, respiration, fermentation, the thing that is needed by organisms to function is adenosine triphosphate, and that is the currency of cells to drive other metabolic processes. So when I said that there are catabolic processes occurring when there are anabolic processes, the currency that we use, the thing that we break down by catabolism to drive metabolic processes, is adenosine triphosphate. Okay, so this was super short, super fast, I know, and there's a whole lot more about metabolism in your textbook. If you want to look at that, that's great, but these are the key points that I want you to understand, as it's going to be important in later chapters. It's going to make more sense then. But this is a very short presentation. Only familiarize you with key points. Please read your text for any materials you don't understand, and I do recommend working the materials available at the text website. As always, if you have any questions on the content, you can post them on the questions and concerns portion of the website. Hopefully, this will help you. Thank you much.